early life experiences are very crucial in the overall organism's development, including development of neurocircuits responsible for the emotionality and cognition. It is well known, well documented from the clinical cohorts that adverse experiences during this early life may impact social, emotional, and cognitive score, and it can, in some cases, it can lead to early death also. So it is evident from the graph that the trajectory of aging with in the individuals with early life stress looks different from the healthy aging. But however, the cellular molecular readouts responsible for this are not clearly understood. So we ask the question, we know early life stress causes accelerated aging, but how that happens is not known. So we ask the question, what is the contribution of inflammation and mitochondria in early life stress evoke accelerated aging? So this we are studying in brain. Particularly for this talk, I will be showing data for the hippocampus because we will be talking about the cognition and we will be dealing with the periphery also. So how do we do this in rodent model? So we have a rodent model of maternal separation as a proxy for early life adversity. We separate the newly born pups away from the mother for initial 2 weeks for 3 hours daily. So this model has been shown to replicate the clinical findings of accelerated aging and mood related disorders. So we allow these separated pups to grow older till their adulthood or middle age. So adulthood in rats, this is done in Sprague Dawley rats. In rats is like 6 to 7 months, we consider adulthood, and 13 to 16 months is like middle age. So in human context, it is like 35 years, 30 to 35 years is adulthood, and 55 years is a middle age. Then we look at the various mitochondrial readouts, neuroinflammation, the central inflammation, as well as peripheral inflammation, then the blood-brain barrier integrity, and the cognitive score. So we started with the peripheral inflammation. So we have the maternally separated rats and we allow them to grow older till adulthood, and we analyze their serum for the circulating inflammatory markers. So some of the inflammatory markers like IL-1 beta, IL-18 showed increase in the levels of these cytokines. Whether they are males or female, we saw the increase. Apart from that, we saw increase in C-reactive protein, GDF15, which is a biomarker for aging, and again white blood cells, WBC count also there is upregulation. When we go to the middle age, we see a prominent phenotype. So everything is upregulated over here in terms of pro-inflammatory cytokines, IL-1 beta, IL-6, TNF-alpha, we have done battery of it. If you look at the anti-inflammatory cytokine, you will see a decline, indicating of inflammaging. We also looked at, as I mentioned, in the middle age cohort, we looked at the CRP GDF15. Again, we saw a persistent inflammation at that point. At similar age, we looked at the neuroinflammation. So we counted the activated microglia in the hippocampus of the brain from the control as well as the maternally separated animals. In this case, it is middle-aged animals. You can see there is, depending upon the morphology, we can calculate the activated microglia. So you can see across the hippocampus, there is increase in activated microglia count, indicating there is more neuroinflammation. So if you compare that with the adult data, in adulthood, you really don't see much of the differences. It only happens in the middle age. So currently we have a data where peripheral inflammation is setting up, but neural inflammation is, it's like peripheral inflammation precedes the neuroinflammation in this case. So we were interested in mitochondria also because mitochondria inflammation can go hand in hand. So we isolated the mitochondria from hippocampus of these animals and looked at the mitochondrial respiration. You can see it shows the different curve. There is an altered mitochondrial respiration. See, if we record the mitochondrial ROS levels at 15 months, which is middle-aged animal, there is higher ROS levels and lower cellular ATP levels. But if you look at the 6 months data, which is adulthood, you don't really see any difference at that stage in the brain. That makes us look at the blood-brain barrier because peripheral inflammation is preceding the neuroinflammation. So we carried out simpler assay using Evans blue. So Evans blue generally does not cross the blood-brain barrier, but if the barrier is leaky, it will cross, and we can count the extravasated Evans blue. So you can see in the middle-aged animals, these maternally separated animals only show extravasated Evans blue, but the adult animals do not show that phenotype. We confirm using radio nuclide assay also, where the technetium-99, you can see the uptake of, there is a radioactivity in this case in maternally separated animals, but in control it is not there. We also perform the gene expression analysis supporting the blood-brain barrier data. Only happens in middle age, but not in the adulthood. So next we done the cognitive score. This is a novel object recognition task to understand their cognitive score. So we give two objects to the rodent to explore like this, and on the second day we replace one of the novel familiar object with the novel object and calculate the exploratory score. That gives us the discrimination index. So discrimination index towards 0 or below 0 indicates there is a cognitive decline. So if you see males or females or short-term or long-term memory retention, you will see the maternally separated animals show cognitive decline. Now, if we focus on the last figure, you see, suppose you focus on maternally separated cohort, you see a distribution over there. Some animals are, they are showing cognitive decline, but some are performing better at that. To understand this, we did the cross correlation analysis. So we did the novel object recognition task and compared with the circulating cytokines profiling. So in maternally separated animals, there is a cross correlation with the the cognitive score is cross correlated with the inflammatory load. To understand this better, the blue line indicates the control animals where we don't see any correlation. But in the maternally separated animals, the score is negative. That means cognitive decline is there. In that case, we see more of higher inflammatory load. So the first part says that if we have maternal separation in rodents, then most of the central phenotype you can see in the middle age, but the peripheral phenotype you can see much earlier in the adulthood. And the cognitive decline is associated with the inflammatory load. Now the main question was whether can we reverse this using some of the strategy. We thought of many and decided on nutritional as well as metabolic modulator, which is nicotinamide. So nicotinamide is a amide form of vitamin B3 and it is involved in NAD plus biosynthesis pathway. So NAD plus is involved in many of the cellular and physiological processes. Secondly, nicotinamide is a known mitochondrial booster. It can dampen the inflammation and it is important in maintaining the blood-brain barrier. See, all in all, it's a good molecule for us to study in this case. So we supplemented nicotinamide in drinking water from late adulthood till middle age in control as well as maternally separated animals. So this become 4-group experiment. And we studied all the parameters which we studied in the first part. So first the object recognition task. As you can see, we have seen the maternally separated animals show cognitive decline, but focus on the fourth, the maternally separated animals receiving nicotinamide, they perform better on the cognitive task. So there is a statistical difference over there. Then we looked at the peripheral inflammation. In this case, CRP, C-reactive protein, which was upregulated in maternally separated animals. With nicotinamide, there is a rescue. Even the GDF-15 levels, growth-derived factor 15. And this is methyl glyxol, so it is involved in advanced glycation end products. That is also coming down with the nicotinamide treatment. We also carried out many other cytokines. Most of the cytokines showed the similar profile. Then we looked at we confirmed that whether nicotinamide is working or not. It showed more of NAD plus levels, so indicating that NAD plus level. So NAD plus levels with aging also itself goes down. So with nicotinamide treatment, there was increase in NAD plus levels. Interestingly, we looked at one of the markers, which is CCF-mtDNA, circulating cell-free mitochondrial DNA. So that is often upregulated upon stress or aging. With nicotinamide treatment, there is a rescue in this. Also, we looked at corticosteroid. So with stress, the corticosteroids goes up. So with nicotinamide, we could see a partial rescue over there as well. We studied neuroinflammation after studying the peripheral inflammation. Again, we did the microglia activation. You will appreciate across the subfields of hippocampus, you can see there is less activation of microglia, which is comparable to the controls in nicotinamide treated maternally separated animals. These are the images for that. Then we studied cellular ATP levels and ROS levels. I said nicotinamide is a mitochondrial booster. So we saw that the mitochondrial ROS levels, which were upregulated here in maternally separated cohort, with nicotinamide supplementation, we could see rescue there, as well as the cellular ATP levels were also mitigated. Then we talked about the blood-brain barrier in the first part. So if you compare the second panel versus fourth panel, the gene expression profile was down in maternally separated animals, but with nicotinamide treatment, we could see rescue in those also. This is a confirmation, imaging confirmation for that. The radionucleotide activities was only seen in the maternally separated animals. Surprisingly, and it is a good indication that maternally separated animals with nicotinamide at this middle age do not show any barrier breach. What we want to do next? So this is about rodent study. So we want to study whether really this holds true in humans, because we don't see any sexual dimorphism in rodents, but it is highly expected in humans. We don't know what will happen. So we are collaborating with the NIMHANS team to do this analysis further. So this is our team, and this was incomplete without the teamwork, and we have excellent collaborators as well. Thank you for your time, and happy to take any question or suggestions. Thank you.
Thank you so much. If you can summarize the hypothesis based on the results in simple terms for people who are not very familiar with these.
So if you have early life adversity, early childhood adversity in humans, you start with third gestation week or till the first decade of your life, sexual, physical, emotional, any kind of adversity, it may result in accelerated aging. What was the mechanism behind that was not known. So we see that the cognitive decline, which is seen later in life, is correlated with higher inflammation. And with the nicotinamide treatment during the adulthood till the middle age, we could rescue this cognitive decline and mitigate the inflammation.
Hi, Pratik.
Yes.
Great talk. So my question is, you didn't see any sexual dimorphism at all?
No.
Like at none of the levels, nothing?
We
That's
have
very
not
interesting.
seen, yeah.
And you're trying to replicate that in a human cohort?
Yes.
Okay.
We think because the model is setting up before the puberty, the stress is between P2 to P14. Okay. And the puberty comes after P45, P50. So maybe that is, we don't know what will happen in human, but till rodent data says that there is no sexual dimorphism.
Okay, thank you.
Is there a quick question there at the back? Yeah, please go ahead.
Yeah, thank you for the talk. It was very interesting. I just wanted to ask something a little more technical about the experimental design. Was there any, the mice pups, especially the ones that were maternally separated, would they grow in a socially enriched environment?
No. So they were separated for 3 hours daily for that 2 weeks, and they were with mother till the weaning period, and then they were in a group of 3 till the age.
So there was no parental figure, they were like not with mother, not with any other.
So it's a uniparental model. So they live with their mother till first month, you can say. Then they get weaning. They get separated from the mother because they can again breed back with the mother. So that is the case.
So actually there are questions. I myself have a question. I'm going to stop because it's a very interesting talk, and sorry to the others who I couldn't come to, but we have to move ahead in the interest of time. So let's thank Dr. Pratik again.
Thank you. We can chat elsewhere also. Thank you.